of our last lecture unit on immunology or host defenses. There's two PowerPoints. This first one, PowerPoint 1, is on innate or nonspecific host defenses or immunity, and the second one is on specific adaptive host immunity or defenses, focusing on humoral immunity and cell-mediated immunity. So just really quickly, folks, there are additional files in the immunology host defenses module that could be helpful. So there is a study guide that also has supplemental notes with it that might be of help. For this PowerPoint, there's a file on kind of a big overview of nonspecific innate defenses. And then there's also a PDF, a scan from a immunology book describing functions of antibodies, primary and secondary immune response, and the key role of T helper lymphocytes, which will be a big topic on the second PowerPoint. Furthermore, our previous micro instructor, Audrey, prepared a document giving you some good links for learning immunology. I think she has some Khan Academy links there. And in addition, there's a practice host defenses immunity quiz also on in the immunology module, so those might help. From last semester, fall 2019, there's a couple of in-lecture movies or videos, so if those might also be of some help. And remember, on those videos, you can go to the settings and you can speed it up to play it doubly as fast, and that might make it a little bit more efficient for you. Okay, so folks, let's see if we can't get started here. I'll try to stick to the PowerPoint slide so I don't go off topic too much. Okay, so innate host defenses or immunity. So we know that we're surrounded by microbes, right? Even in lab when we did the, or had we been there when we did the airborne microbe experiment, we saw that even our air is filled with microbes, or on every surface. So we might ask the question, why aren't we constantly infected by microbes, right? If we're basically being bathed in them all the time. So we want to remember that animals, organisms, cellular organisms, we've been co-evolving with microbes, right? So there's been natural selection, so animals and humans have evolved structures and chemicals for protection against the microbes. And very often, historically, folks, we talk about immunity or host defenses providing protection against invading microbes. So there's often a lot of like warfare or battle vocabulary that's used. And me being just kind of goofy, I was thinking, what would be a good analogy? So I was thinking of the Lord of the Rings, and my goofy analogy, you guys, was when the orcs, who were the pathogens, who we'll say are the pathogens, they cause suffering, attack the peoples in Helm's Deep, right? And so if we look at the defenses of Helm's Deep, first of all, there's this outer wall, these thick, strong outer walls that would prevent the orc pathogens from invading. So we're gonna use this as our example of surface defenses provided by skin and mucous membranes. But then, unfortunately, remember, the orcs blew a hole in the wall, so that would almost be maybe like a nail or a stick or maybe a syringe or a bite. So the pathogens, they break through the surface defenses, break through the walls, and they get inside the body. And so inside the body, there's another level of nonspecific innate defenses, and we'll just call these the innate interior defenses. And we're going to see that there's going to be cells, like the soldiers here fighting, that are instantly activated, that are trying to protect us against the invading pathogens. So these could be like our neutrophils, perhaps. And then there's going to be some chemical defenses as well. What we'll see is one of the good things about innate surface and innate interior defenses is they protect us against a wide range of pathogens. It's not specific. And another thing that's really important is that they're instantly activated, right? They're always there. They're always ready to go. But the problem is, especially with microbes that have several virulence factors, sometimes they can overwhelm these innate nonspecific defenses. So then we really need big help. I think of the third layer of defenses, adaptive, specific defenses or immunity. This is all like the cavalry, or this is Gandalf arising, right? With the riders that are going to sweep down and just knock out all those pathogens. So this specific adaptive host defenses or immunity will be the topic of the second PowerPoint. And again, you guys are going to talk about the power of antibodies in protecting us, and then cell-mediated immunity. In this one, we're going to talk about innate surface and innate interior defenses. So just some key points, folks, on innate nonspecific defenses. Reset, a huge advantage is that they're always ready to go, so their response is really fast. All eukaryotes have some form of innate defenses. It is possible that they may kill invaders, and if not, hopefully innate defenses are slowing the invasion of the pathogens down so we can activate those slower adaptive defenses. Nonspecific in these innate defenses are effective against a wide range of microbes. The response, as we said, is nonspecific, so it's not tailored for a specific invader or, say, a toxin. And this is a huge disadvantage, folks. So there's no immunological memory in innate nonspecific defenses. So the second time we encounter the same pathogen, there'll be no amplified response, no greater response, because there's no memory cells. And we'll see that in the adaptive specific immune responses, the advantage of those responses is they do have memory cells, right? So there'll be a stronger, faster, longer lasting, more effective, what we call secondary immune response in adaptive immunity, but we don't get that with innate defenses. So folks, I'll let you look at it. This is nice. It's a list of all the first line defenses, physical, mechanical, and chemical defenses, but I'm just going to go ahead and walk through our slides to give us some examples. Okay, so first of all, folks, a really, really important barrier, of course, is our skin. So one of the primary functions of skin is to prevent the invasion of microbes, right? So it's a physical barrier, right? The deeper layers of skins, the cells are tightly attached to make it impossible for the microbes to sneak between the cells. Healthy skin, folks, should be relatively dry, and it should have a relatively low pH. So a lot of microbes require water just like we do. So with the skin being dry, that will inhibit their growth. And the low pH will also inhibit the growth of a lot of microbes. Our good old normal members of the microbiome, I should update this to microbiome. So we have a lot of commensals, some mutualists that live on our skin, and they are part of our natural defenses. They're going to block attachment sites of potential pathogens. They'll compete not only for space, they'll compete for nutrients. And another good thing about members of the normal skin microbiome is they produce metabolic waste products like acids that can inhibit potential pathogens. So always remember that, folks. Our normal microbiome, it is part of our natural defenses. We make salty sweat for evaporative cooling, but that sweat also can help create kind of a hyperosmotic environment, right, to help inhibit the growth of microbes. We also make lysozyme. This is the enzyme that will hydrolyze peptidoglycan, right? We make fatty acids, and these again can be inhibitory to some microbes. And this sloughing of the cells, you guys, up here, this layer of cells right here are actually dead cells. These are the okay, help me out here, you guys. Stratified squamous keratinized epithelial cells, right? So as those epithelial cells mature, they become filled with this tough antimicrobial protein called keratin, right? And that keratin provides strength here. And the keratin itself is supposed to be antimicrobial. And then furthermore, folks, like every day we shed this top layer of dead cells, right? They slough off, they fall off. And so if we have any potential pathogens attached to that top layer of keratinized cells, they'll just literally be physically removed when they fall off of us, right? Now, one thing that is, you could argue a little bit of a weakness here, folks. This is a hair follicle, right? So there'd be a hair extending out here. Staphylococcus aureus is really brilliant at growing down into this hair follicle. And often down towards the bottom, they can form a little abscess here, right? So very often you'll have little abscesses, maybe like little pimples that are associated with hair follicles where Staph aureus has grown deeper. The Staph aureus hasn't broken across, right, that surface layer, but it's just permitted them to grow more deeply, right, than if they were just up here. And that would protect them from being sloughed off, right, by that layer of dead keratinized cells. And then folks will next look at mucous membranes. So mucous membranes have kind of a tougher job because they're continually being exposed to microbes from the environment. But because of their function, they can't be dry. They can't have that keratinized layer of protective cells. So instead, what's really cool with mucous membranes is that they're cells, I believe they're called goblet cells, that will secrete this sticky mucus blanket that covers the mucous membranes, right? And I always think of that as kind of microbial flypaper, right? So the microbes that come into contact with their mucous membranes, maybe the conjunctiva of the eye, of our respiratory tract, the gastrointestinal tract, the genital tract, hopefully a lot of those potential pathogens would get stuck in this mucus blanket, right? And then we'll see there's different methods that we can use to get rid of the mucus blanket. If it's in the respiratory tract, we could cough or sneeze out the mucus blanket, or we could if we have an accumulation of mucus in the back of the throat, right, we could swallow it, and then that would cause death of the microbes in the low pH of the stomach. We could spit it out, right? If we have microbes, say, in the bladder or the urethra, stuck in this mucus blanket, when we urinate, we could physically remove them. Likewise, microbes stuck in the mucus blanket of the intestinal tract, right? We're going to defecate, we're going to physically remove them. Furthermore, folks, on the mucous membranes, the top layer of cells eventually will fall off, right? So if we have microbes attached to them, they'll be sloughed off and then physically removed. Once again, you guys, we can't stress enough how important members of the normal microbiota is a little bit of um, an older term, but good members of the normal microbiome, the commensals and mutualists, they play such an important role in protecting us against potential pathogens. In that sticky mucus blanket, there'll be lysozyme, that hydrous peptidoglycan. And then, folks, we've already introduced you to this when we were talking about damage caused by viral infections. We've introduced you to the concept of the ciliated cells in the respiratory tract, and we'll also see, folks, on one of the following slides, we'll discuss the ciliated cells of the oviducts or fallopian tubes in women. So just a quick review, you guys, of the upper respiratory ciliated cells. So sometimes this is referred to as a mucociliary escalator. So here are these beautiful long, fine cilia, the ciliated cells of the upper respiratory tract. I think these are the goblet cells that have secreted this sticky mucus blanket here, right? So this sticky mucus blanket is where the microbes and any other garbage we breathe in would get trapped. And then these beautiful ciliated cells, they beat all together to move that mucus blanket with the entrapped microbes up to the back of the throat, where we can either swallow it to destroy the microbes in our stomach, or we could spit it out, right, to physically get rid of the microorganism. So the mucociliary escalator in the upper respiratory tract is really crucial for protecting us against inhaled microbes that could cause potentially, if we let them get down into the lower respiratory tract, they could cause really serious pneumonias. And indeed, you guys, with the COVID, the SARS coronavirus 2 that's causing the present pandemic, the viruses have somehow escaped being removed by the mucociliary escalator, because we know that they're causing really serious pneumonia. Okay, so one thing that could damage the mucociliary escalator is chronic smoking. So folks that have smoked for many, many years, they might be at higher risk for respiratory tract infections, and especially for lower respiratory tract infections, pneumonia. Folks who have careers in which they're working around a lot of smoke, so folks that are firefighters, for example, would be at higher risk for damage to the mucociliary escalator, right? And it's of interest during like really, really cold weather. My understanding is the mucociliary escalator doesn't work as well when it's really, really cold out, and that might be one part of the puzzle why there's more respiratory infections during wintertime. Okay, so any kind of damage to the mucociliary escalator is going to increase our risk of respiratory tract infections. Just a cute, incredible electron micrograph. You guys have those beautiful ciliated cells. And then, folks, what I'll do is just staying on that same track, you guys. We'll talk about how there's another place in humans where we have ciliated cells, kind of the equivalent of a mucociliary escalator, and that's in the in females, in the oviducts or fallopian tubes. We have ciliated epithelial cells in the fallopian tubes or the oviducts. So um, not only is this part of our defenses, but these ciliated cells are really important to help move the egg, the ovum rather. So approximately once a month, a mature woman would release a single egg or ovum from the ovaries. And the problem is that in mammals, our eggs don't have any structures for motility. They don't have flagella, they don't have cilia. So the question is, how would the egg move from being released from the ovary? The egg has to make this journey. It has to enter the oviduct or fallopian tube. It has to travel along, and then it has to enter the uterus, right? Where if the egg is fertilized, right, the fertilized egg would implant in the uterus wall, and that's where the baby would develop. If it's not fertilized, right, the egg would be shed out through the cervix and through the vagina. But again, you guys, mammalian eggs lack cilia or flagella. They're non-motile. So one really important job of ciliated cells in the oviducts is to beat and create currents that will passively move the egg through the oviducts and then into the uterus, right? Now, there are huge problems in women who have what we might call silent genital tract infections, and two notorious pathogens that can cause a so-called silent genital tract infection in women are the two bacteria, chlamydia and Neisseria gonorrhea. So what can happen is that if a woman becomes infected with Neisseria gonorrhea or chlamydia, we can have an ascending infection. The microbes can move up here, and then they can move into the oviducts, the fallopian tubes. And here they can cause chronic inflammation, which will damage those ciliated cells. And indeed, in severe chronic infection, you might even have fibrosis here. Now, why is that a problem? Because if the woman ovulates, if she releases the egg, maybe the ciliated cells here at this this end of the fallopian tube are still working. So the egg maybe gets drawn up here, but then when the egg hits that area of damage caused by the Neisseria gonorrhea or the chlamydia, these ciliated cells are damaged. So basically, the egg gets stuck there in the oviduct or fallopian tube. And then you might say, well, that shouldn't be a big deal, is it? But the problem is, folks, if the woman is inseminated, right, the sperm have flagella, and the sperm are really motile. So if the woman is inseminated with sperm, the sperm, they're like racehorses. They're trying to find that egg, and they can move up here. And if they find the egg that's stuck in the fallopian tube or oviduct, the sperm will still fertilize the egg. But now that fertilized egg can't move down to the uterus where it needs to move to, right? So this is called an ectopic pregnancy, a pregnancy that's occurring in not the correct space, right? So you can imagine, folks, this could be life-threatening, right? If the fertilized egg, the fetus is developing in the fallopian tube, right? The fetus isn't going to make it. We can have damage to the tubes that could result in hemorrhage. So this is really serious condition for the mother as well. So this is one reason, you guys, it's so important that women, especially if they're sexually active, that you get at least, annual exams, right? And you want to, if you're sexually active, you want to make sure that you're being screened for sexually transmitted pathogens, because you don't want to end up with one of these silent infections. So we know that these chronic infections, untreated infections with chlamydia and Neisseria gonorrhea, they can cause ectopic pregnancies. We can have massive chronic infection here that leads to PID, pelvic inflammatory disease, right? It's been, in some cases, the damage might be so severe that it might lead to infertility later in life. The woman can't conceive later in life. And furthermore, you guys, having chronic infection in the genital tract will increase the risk for HIV because you're basically attracting the white blood cells that HIV wants to infect, right? So again, these chronic, long-lasting sexually transmitted infections, we have damage to the ciliated cells of the oviduct. It's increased risk of pelvic inflammatory disease, increased risk of HIV infections, ectopic pregnancies, that was left out here, you guys, sorry, and then the possibility of infertility. All right. So, and with both Neisseria gonorrhea and chlamydia, folks, they're bacteria, right? So you can be treated with antibiotics, right? But just need to make sure that the treatment starts early on. Okay, so now you guys will just back up and kind of go a little bit more general surface defenses of the genital tract. So again, we have these wonderful mucus blankets. The ciliate epithelial cells in women kind of act like that mucociliary escalator, because if microbes can get stuck in the mucus blanket, maybe up here, right, the ciliated cells can move it here to the uterus, and maybe that microbial flypaper then could exit through the cervix and the vagina. Men have like a combination of urinary tract and genital tract. So the urethra, when men urinate, that's going to help flush out any pathogens that have infected the urethra. In women, the vaginal mucosa is often colonized by our good lactic acid bacteria, right? Good members, mutualists of our normal microbiome. The lactic acid bacteria, they make lactic acid, and that's going to inhibit a lot of potential pathogens, right? Oh, and this little bit here, folks, is just to remind me that often when men are infected, say with Neisseria gonorrhea or chlamydia, because the urethra is being infected, when the urethra becomes inflamed, it'll become really, really painful to urinate. So very often, men who are infected with Neisseria gonorrhea or chlamydia, they're symptomatic, right? They're going to have that burning sensation when they urinate, so hopefully they'd run to the doctor, right, to be treated with antibiotics. But in women, unfortunately, the urethra here leading up to the bladder, and then this is, I have my anatomy right, you guys yell at me. So this is the uterus and the cervix and the vagina. Sometimes a woman can have a genital tract infection of the reproductive tract without the urethra getting infected. So maybe she's not having a burning sensation when she urinates, right? We want to have those clinical signs and symptoms so that we can go get medical treatment, get antibiotic therapy. And this is just more detail, you guys, these beautiful ciliated cells to move the egg towards the uterus, and also the ciliated cells will move the microbes away from the ovaries and the oviduct. And again, this is just the concern with silent genital tract infections that can damage the cilia, lead to ectopic pregnancies, sterility, increased chance of other infections, and also I left out here pelvic inflammatory disease, PID. So I'll try to do this really fast, you guys. So the digestive tract, the urinary tract, they all have mucous membranes, right? In the digestive tract, we have low pH. I think it's around pH 2 in the stomach, and that's going to destroy a lot of pathogens. But especially if the pathogen is protected, say in food, we know that there are like fecal oral pathogens that can survive passage through the stomach. They reach the intestine, so we have bile that can help destroy a lot of microbes that haven't evolved to live in the intestine. Thank heavens for the motility, right, in the digestive tract, because the movement of food and eventually feces can physically help get rid of a lot of pathogens. And again, you guys, we can't overemphasize the protective, defensive nature of our good normal microbiome. Urinary tract, really important, you guys, that when you need to urinate, when you need to pee, go pee, because that flushing action is helping to wash out microbes that are ascending up the urethra, right? So ideally, you never want to try to hold your bladder. Again, we have a nice mucus blanket that would be removed with all the stuck microbes as we're urinating. But you can imagine, you guys, what happens when we insert urinary catheters, right? The tip of the urinary catheters, although there's supposed to be new designs to prevent this, but we'll just pretend like the tip of the urinary catheter is being inserted up here. You can imagine it becoming contaminated, maybe with microbes from the skin in women. I mean, you could have like fecal microbes down around the opening of the urethra. And then as you're inserting the catheter, you're actually inserting the microbes up into the bladder. So very often, unfortunately, sequelae to having a urinary catheter put in place is you end up with a bacterial cystitis, right? And for something like that, I think you almost have to remove the catheter, right, and then treat with antibiotics. So folks, we're going to pretend that our surface layer of defenses, the walls of Helm's Deep have been breached. Here are the orcs, penetrating. Okay, so here we have like a sliver, a little piece of wood that, of course, has microbes on it. So in this picture, you guys see a little green bacilli here. These are the invading microbes. So we've punctured through surface defenses. So the question is, do we have any defenders here in the interior that are part of innate nonspecific defenses? And the answer is yes. So what we're going to do, you guys, we're just going to look at some of the interior nonspecific defenses. We'll look at inflammation. I think I reverse order here, you guys. Phagocytosis, fever, complement activation, and interferon. So these would be like maybe like the soldiers inside Helm's Deep. They're just going to battle the orcs as soon as they come in. So folks, inflammation is our response to any kind of damage. It doesn't have to be microbial. If we were in lecture, what I do is I would just like scratch my wrist with my fingernail, just causing physical damage. And we would see within minutes, we'd see some of the signs of inflammation. So some of the classic signs of inflammation are redness, swelling, pain, and heat. And so what we want to do is walk through why we're seeing these changes, right, and how these changes are being triggered. So if we did it just really quickly, you guys, the reason the area that's been damaged will appear red is you have vasodilation. The blood vessels get bigger, so more blood is moving through the area, and blood has red blood cells, so the area will appear red. The reason we're going to get the heat is also because we have more blood moving through the area. So we'll have an increase in temperature. That'll be the heat. And oh my gosh, I've been so amazed. Some physical therapists, they're so sensitive, like is they're maybe examining a joint, they can feel that increase in heat. They're that sensitive, right? And again, that's a sign of inflammation. In addition to blood vessels undergoing vasodilation, getting bigger, the blood vessels become leaky. So we start losing the liquid portion of the blood, the plasma, into the extravascular spaces, the areas outside the blood vessels. And that's what's going to cause the swelling or the edema, right? And then in response to this damage, we're going to see that cells of the innate immune system will release chemical messengers, and those chemical messengers can interact with pain receptors, right? And then we'll interpret those pain receptor impulses as being painful, right? And that makes sense, you guys, because if we have like a stick rammed into us here, we want to know it so we can pull it out, right? So pain, it's evolved to let us know something's wrong. We need to change something, like, for example, pull the stick out of your foot, right? Okay. So then, folks, this is really simplified kind of steps in inflammation. So let's just walk through it, okay? So when there's any kind of tissue damage, including microbial invasion here, it's gonna trigger our cells to release these chemical messengers. And you guys, we're just gonna call them inflammatory mediators, chemical messengers, which are gonna initiate the domino effect for all the changes we'll see in inflammation. So in response to these inflammatory mediators, and probably the most famous one, you guys, is histamine, right? We're going to see all these changes. So in response to the inflammatory mediators, the blood vessels dilate and they become leaky. So the dilated vessels, more blood, the area appears red, they become leaky. This is what will contribute to the swelling or edema, right? And this is so cool, you guys. So the cells making up the blood vessel walls, these little endothelial cells. So here's a blood vessel, you guys, right here. a little capillary, very delicate little capillary. These cells making up the walls of the vessels or the capillary are called endothelial cells. So in response to these inflammatory mediators, the endothelial cells are going to start making um, um, surface molecules to which our white blood cells will attach. This is so cool. So what we want in inflammation is we want to deliver all these cells of the immune system that will help destroy the invading microbe, as well as some antimicrobial chemicals we want, right? So the question is, well, how would the white blood cells, and you guys will use as example our phagocytic cells. So our first responders when we have any kind of tissue damage are neutrophils. They're the fastest. They're relatively short-lived, but they're the fastest. If they get there really fast, they could ingest, destroy the invading microbes and no problem. And then a second phagocytic cell you guys will be talking about are macrophages. They're slower than neutrophils. So they get there a little bit slower, but they're much longer lived. And furthermore, you guys, we're going to see they can help trigger adaptive specific immune responses. Okay, so how would those, for example, neutrophils know where the invasion is? Well, remember how the endothelial cells express new surface molecules. In response to those inflammatory mediators, the neutrophils are going to express new molecules that can bind to the molecules on the endothelial cells. So maybe, I hope this will work, guys. We could say the neutrophils will express new receptors that can bind to the endothelial cells of the vessels, right? So that will get the white blood cells to start binding to the walls or the margins of the vessels. So that process is called margination. They can actually see this. They can do experiments with vessels and actually see the white blood cells marginating. And then you guys, this is so amazingly cool. The endothelial cells are going to retract a little bit, just pull in a little bit. That's what makes the vessels leaky, right? So not only are we leaking out the liquid portion, the plasma portion with antimicrobial chemicals, but by retracting, that's going to make it easier for the white blood cells to squeeze between the endothelial cells. Isn't that amazing? So in the old days, they would call that process, so we had margination and then the white blood cells squeeze between the endothelial cells. In the old days, they would call that diapedesis. I think nowadays they have a fancier term like transmigration, right? So now the white blood cells are, they're no longer in the blood vessel, but still, how are they going to find those invading microbes? So remember all those inflammatory mediators. And in addition, you guys, the microbes are going to be releasing microbial substances. That's going to set up a chemical concentration gradient. And so the white blood cells can use that chemical concentration gradient to guide their movement towards ground zero, right? So that process, you guys remember, is called chemotaxis. Isn't that amazing? So then, again, going back, and let's say these are neutrophils, right? So they use chemotaxis to arrive at ground zero where all the microbes are invading. So then hopefully the neutrophils can attach to the invading microbes, ingest them, and kill them in the process called phagocytosis, right? So if we were going to look at some of the advantages of inflammation, we're going to increase delivery of blood. That's going to increase delivery of the leukocytes, for example, the neutrophils or phagocytic cells. It will also increase the delivery of antimicrobial chemicals, which we will be discussing here in a few slides. It's going to attract and activate the leukocytes. It's going to guide them to the site of invasion. And what we might say, you guys, is one of the goals of inflammation is to try to maybe trap the invading microbes here and hopefully kill them. And if not kill them, at least maybe try to trap them so they can't spread. And try to maybe keep the invading microbes in lower numbers. And this is really important, you guys, because we're going to see our big guns, Gandalf and the riders. It takes them a while to get to the fort that's under attack, right? So indeed, it's the same way, you guys, with adaptive specific immune responses. It's going to take about 10 days before those powerful defenses arrive. So if nothing else, maybe innate defenses can at least kind of corner the pathogens and prevent them from spreading very far, right? So folks, this was from our previous micro book, so I'm going to let you read this. It's got a lot more details. It talks about the chemical messengers, but generally speaking, you guys, the questions on um, our immunology questions on the exam will follow um, more of the slides, and certainly, folks, your best guide is going to be your study guide. So your immunology study guide, that's the place you want to go. Okay, and there are some additional notes, kind of key points in the study guide. All right. Oh, and again, folks, we do want to remember that part of innate defenses, we could end up with pus being produced, right? So remember good old Staphylococcus aureus and Streptococcus pyogenes. Pyogens are pus producers, right? They attract white blood cells, and especially if the pathogens can produce leukocytes, are going to end up killing a lot of the white blood cells. And we'll have this lovely, thick, kind of creamy white, creamy yellow pus filled with dead and dying white blood cells and the pathogens and other cells from the host. This, folks, I think is really important, and you'll get tired of me saying this, but I'm going to say it anyway. So folks, our immune system is often described as a double-edged sword, meaning there's two sides of the swords that can cut. So let's say that you have a sword. It's in the old days, right? And the enemy is attacking. The orcs are attacking you, right? So you're using your sword maybe to try to hit the invading pathogen to hurt it, right? Because ideally you want to kill the invading pathogen before they kill you. So one side of the sword is to kill the invading pathogens. But let's say you maybe, I don't know, you strike with so much force, you kind of get this rebound. The sword bounces back, and the opposite side is also sharp, and you end up cutting yourself. So that's how I see the immune system. We can't live without it, right? We need it so we can defeat, kill invading pathogens. But our immune responses themselves can cause a lot of damage. And a perfect example, you guys, is chronic inflammation. When you have an inflammatory response where it just keeps going on and on and on and on, and we're going to see, and we'll be using our phagocytes as an example. Phagocytes can be really destructive. They can release chemicals that are going to cause damage to our tissues, right? The chemicals are meant to kill the pathogens, but the problem is sometimes the phagocytes release the chemicals and they actually damage our own tissues. And folks, there's so much interest in chronic inflammation. They think it is a cause for vascular disease like atherosclerosis. It's thought that some cancers involve chronic inflammation. Um, Progressive neurodegenerative diseases such as multiple sclerosis and Alzheimer's disease um, may very well involve chronic inflammation. It's fascinating, you guys, because Dr. Holland pointed out that some of the risk factors for really serious COVID-19, the pandemic coronavirus infections, like they talk about diabetes and heart or vascular disease, and I'm thinking there's one more, but she pointed out all of those conditions involve chronic inflammation, right? So again, chronic inflammation is not good for us. Yeah, and it can lead to all these other diseases. Folks, this was to go back actually to our medical micro unit. And do you remember when we were talking about bacterial toxins, we were talking about the protein bacterial exotoxins, and then we were talking about the gram-negative bacterial endotoxins, right? And the endotoxin is part of the cell wall of the gram-negative bacteria, right? So it's usually not secreted in large amounts while the bacteria are alive, but when they start dying, they start breaking down, their cell walls break apart, they're going to release a lot of LPS without lipid A. And what happens is the lipid A portion of lipopolysaccharide will bind to cells of our immune system. And if we have a lot of endotoxin, the lipid A of LPS released, then we have excess of those inflammatory mediators. Or another term, you guys, we could use for these inflammatory mediators, the chemical messengers that our white blood cells release, we could call them cytokines. So The problem when we have gram-negative bacteria growing in our bodies where they shouldn't be, like in the blood or the central nervous system or in the peritoneal cavity, we get massive release of endotoxin. We get massive release of these chemical messengers, these cytokines from white blood cells. And this can kill us. This is often referred to as a cytokine storm. This is very often you hear this referred to as gram-negative bacterial sepsis or septic shock. Rapidly, you guys, we can die from this. So it's like, what the heck's going on? Well, if we think about it kind of from an evolutionary point of view, inflammation is good if it's local and short-lived, right? You want to quickly trigger inflammation so you can kill the invading pathogen. But if you have this massive release of endotoxin and this massive cytokine storm, your whole body is being flooded with these chemical messengers. So instead of just locally having vasodilation and leaky vessels, now all throughout your body, your blood vessels are dilating and becoming leaky. And this is going to be horrible because what will happen is that is the our blood vessels dilate everywhere and become leaky everywhere. That's going to cause a drop in blood pressure, right? And we're losing fluid into the extravascular tissues. The blood vessels are dilated. So that can throw us into what's called hypotensive shock, right? With low blood pressure, we don't get enough blood delivered to our tissues, our organs, right? And they can't survive without blood. And furthermore, you guys, you might remember that we said in response to the cytokine storm, we'll have activation of the clotting cascade. So now we're going to end up with, let me see if I can't get my little cursor here to work, okay. We're going to activate the clotting cascade, coagulation cascade, and as a result, we're going to end up with what's called DIC, disseminated intravascular coagulation. So we're throwing blood clots everywhere, and that's further going to block blood flow, starving our tissues, our organs for oxygen, right? And then the irony of this is, you guys, that we said if DIC continues too long, you use up all your clotting factors, right? So then you start bleeding out. It's so ironic, right? First, you're forming too many blood clots, use up all your clotting factors, and then you're going to start to hemorrhage out, right? Gosh, it's just horrific, right? Now, with endotoxin, a lot of the, um, I shouldn't say a lot, but in response to the endotoxin, white blood cells will release chemicals that will circulate to the brain and cause an increase in body temperature, right? So remember, we said endotoxins are pyrogens, right? And here, folks, and again, you don't have to memorize this this kind of concept map, but it just gives you an idea, you guys, of how many body system, organ systems your poor patients are going through. So there's their liver shutting down, right? We've already talked about the DIC and hemorrhaging. So all the tissues, all the organs are going to be lacking sufficient blood. We end up with kidney failure, right? We have impaired heart function here. And here's the vasodilation increase. Capillary permeability, we have damage to the lungs, right? So, oh, and the brain, right? So often folks in this so-called endotoxic shock, they're confused, right? Because the brain's not getting enough blood, right? So we can end up with respiratory failure, shock, kidney failure, DIC. So very rapidly, the patient can die from this endotoxic shock, this sepsis, septic shock, right? And so even people that can survive this, very often they have like permanent renal damage. One of our distant acquaintances developed septic shock, and even though he survived, he's never been the same, right? It's like there's permanent brain damage done, right? And indeed, some of the survivors, I've read that they're at kind of permanent risk for long-term kidney disease, right? So this is a tough one, you guys. Wouldn't it be awesome if we had a vaccine against endotoxin, right? Maybe one of these days they'll develop technology where we can make a vaccine so we can vaccinate ourselves against the endotoxins. So folks, you'll recall that we said is part of inflammation, part of it was to try to attract and guide phagocytes to the scene of invasion, and their job is to try to engulf and destroy the microbes, right? And the two phagocytes we'll describe, you guys, are neutrophils, sometimes called PMNs for polymorphonuclear leukocytes. They're the EMTs. They're the first to arrive at the scene, maybe within minutes, but they're relatively short-lived, right? And then slower but longer lived are the macrophages. Literally means big eaters. And again, you guys, these guys, they can live a lot longer. And another thing that's really cool is they can help trigger the adaptive specific immune responses. Yeah, very cool. So this has been a really neat area of research, you guys. So it's like, how do cells of the innate immune system, so how do, like, our neutrophils, for example, how do our macrophages, how do they recognize those invading pathogens? So a lot of research has been done on this topic, you guys. So these cells of the innate immune system, and again, we'll just use neutrophils and macrophages, on their surface, they have these really cool, what are called pattern recognition receptors. And this is so neat, you guys. These are receptors that will recognize and bind to unique microbial substances, substances that our bodies don't make, right? Because we don't want them binding to our cells and destroying them, right? So the probably the most famous pattern recognition receptors are called the toll-like receptors. And there's a long and amazing story for their discovery, but they were initially discovered. I think they were doing genetic experiments with fruit flies, and they identified these wild receptors on the fruit fly cells. And in German, toll, to me, when I hear toll in German, it means like cool, wild, radical. So the initial scientists called these toll-like receptors, but we now know they play a really important part of the innate defenses, right? We have these pattern recognition receptors, like toll-like receptors, on the surfaces of our neutrophils, our macrophages, just as an example. And here, you guys, I love this little cartoon because it's showing us there's different toll-like receptors. They're numbered here. Some are on the surface of our cells, some are in the inside, like in endosome or phagosomes, right? But we'll just use the ones that are on the surface of our cells. So what could they bind to? Well, this toll-like receptor 4, TLR4, binds to lipopolysaccharide. Who makes lipopolysaccharide? Only gram-negative bacteria. Here's a toll-like receptor that binds to bacterial flagellin. Do humans make bacterial flagellin? No, right? So that's another way for your phagocytic cells to identify an invading bacterial pathogen. In and they've just done this really generally, you guys, for gram-positive bacteria. Lipoteichoic acids unique to gram-positive bacteria could be recognized by toll-like receptors. In both gram-positive and gram-negatives, peptidoglycan can be recognized by toll-like receptors. And mycobacteria, and I haven't double-checked this, you guys, I didn't do my homework, but I'm wondering if maybe like the mycolic acid might be recognized by a toll-like receptor. Components in the walls of yeast, right, can be recognized by toll-like receptors. Yeah. And then in the toll-like receptors inside, we have toll-like receptors that can identify viral and bacterial nucleic acids because they'll have different little chemical signatures to them, right? So in reading about these pattern recognition receptors, we're hardwired for these. They're part of our genome. So our neutrophils and macrophages, based on the genes they carry, will make certain pattern recognition receptors. And I'm not sure, folks, but maybe this is why, like, some people seem to be really resistant to microbial infections, and others are not. So I'm always interested, like in our family, 3 people in our family could be sick, but the fourth person never gets sick. And I'm like, why is that? And it might be that fourth person just has an awesome array of pattern recognition receptors, so that early on in infection, the pathogen will be destroyed. And maybe the rest of us seem like we're more frequently getting infected, maybe just genetically we don't have as many of these pattern recognition receptors, so we aren't as good as killing the microbes when they first invade us. It's just a thought. So folks, so again, the point here was to discuss steps in phagocytosis, which is just remarkable. And again, folks, we're going to focus on the short-lived, fast neutrophils and the longer-lived, slower macrophages, right? So remember, folks, how do the phagocytes How are they guided to the scene of microbial invasion? And that's going to be through chemotaxis, right? And then using their pattern recognition receptors, for example, the toll-like receptors, they have to bind to the surface of the invading pathogen. If they can't bind to the surface, they can't ingest them. And remember, you guys, that was the problem with bacterial capsules. Our phagocytes never evolved receptors to bind to bacterial capsular polysaccharides. And that's why those encapsulated bacteria, they're slippery. Our phagocytes can't grab onto them. So our phagocytes have to have receptors to be able to bind to surface molecules on the invading pathogen. We'll talk later, you guys, about opsonins. Opsonins, as we mentioned in our immune responses to viruses, little subunit in our viral module, opsonins can coat the invading pathogen, and then the phagocytes have receptors for those opsonins. And so opsonizing the invading pathogen increases phagocytic killing of them. And I'll just mention it really quickly here, you guys. Two opsonins are complement and antibodies. Okay, so once the phagocyte is bound, it'll extend these pseudopods, these little false arms around, and the pseudopods will fuse, and that will form what's called a phagosome. So here's the phagosome. This is the actual endocytosis, phagocytosis. Here's the phagosome formed, okay, or phagocytic vessel. Now the problem is that a lot of microbes are really happy living in the phagosomes, right? So there has to be a really important step, and that important step is that we have another membrane-bound vesicle called a lysosome, literally a lysing body. This lysosome is chock full of hydrolytic enzymes, digestive enzymes. So really important in phagocytic killing, the lysosome has to fuse with the phagosome. And this will form, and again, if you've had German, you guys, you know, you just keep adding words together. This will form the phagolysosome, and this is where the actual microbial killing occurs. Now, this is so neat, you guys. These hydrolytic enzymes inside the lysosome, they're in an inactive state. We could describe them as being acid activated, right? So for those hydrolytic enzymes to change shape into their active conformation, we have to have an acidic pH inside the phagolysosome. How will that be accomplished? Proton pumps. Isn't this amazing, guys? So there's proton pumps that will pump protons from the cytoplasm into the interior of the phagolysosome. That drops the pH. So when the hydrolytic enzymes are introduced into that low pH, they change their shape, right? They refold into their functional conformation, right? So we'll have proteases, nucleases, lipases that hopefully can kill the microbe, right? Just simply digest it. Now, that process of using the hydrolytic enzymes to kill the microbe, that's called oxygen independent killing, because we don't see an increase in oxygen consumption. This is kind of like the first punch of a 1-2 punch, right? If somebody's attacking you and you're trying to punch them in the face, the first punch maybe doesn't knock them out, but maybe the second one will. So the second punch, you guys, in this phagocytic killing is called oxygen dependent killing, because it requires a massive consumption of molecular oxygen. It's sometimes called the oxidative burst or respiratory burst. And this again is really cool, you guys. So in the membrane of the phagolysosome, there's an activated enzyme system that will start making those incredibly powerful toxic reactive oxygen intermediates. And you might remember, you guys, in the microbial growth unit, we were talking about some of those, and some of the reactive oxygen intermediates include superoxide anions or radicals, includes hydrogen peroxide, hydroxyl radicals, and even a bleach-like substance, hypochlorite, can be developed, right? So that's the second punch, those toxic reactive oxygen intermediate superoxide anions, hydrogen peroxide, hydroxyl radicals, and bleach-like hypochlorite substances. So that's the second punch. So hopefully with that 1-2 punch, you guys, we're going to kill the microbe, we're going to digest it, maybe some of the nutrients can be absorbed, and then we're going to have waste here, right? A little waste vesicle. So the phagocyte has to get rid of it. It will migrate over the cell membrane and fuse, and then in the process called exocytosis, dump all of that garbage into the extracellular space, okay? So again, the goal is to try to kill the phagocyte. But again, you guys, there's problems. There's always problems, right? So in this process of exocytosis, what's being dumped into the extracellular environment? Those activated hydrolytic enzymes. All of those reactive oxygen intermediates are getting dumped into our tissues. And this helps us understand, folks, that again, going back to the double-edged sword, right? Why chronic inflammation can cause so much damage, right? We have to kill the microbes, but some of the weapons we use will actually end up hurting us as well. And again, chronic inflammation, you constantly have these phagocytes arriving, trying to kill. They're dumping the reactive oxygen intermediates, dumping the hydrolytic enzymes, and eventually the phagocytes will die, you guys. And then when they die, they lyse, we dump even more of these toxic molecules in the surrounding tissues. And then, folks, there's some uh, microbiology authors that I just love because they're always giving things personalities, which I love to do. So they talk about sloppy feeding phagocyte. So what's a sloppy feeding phagocyte? It's a phagocyte where maybe in its excitement to try to kill the microbe, the lysosome gets confused. And in a sloppy feeding phagocyte, instead of the lysosome fusing with the phagosome, it goes over here and fuses with the cell membrane of the phagocyte. So it dumps all those hydrolytic enzymes again into extracellular space. And when we have chronic inflammation, very often this will be an acidic environment. So again, those acid activated hydrolytic enzymes, they refold, and now they're starting to harm, hydrolyze our tissue. So again, you guys, just this example of the double-edged sword and why chronic inflammation can cause a lot of damage. And then another problem, you guys, if you might, again, remember in the medical microunit, we talked about how some bacteria have evolved ways to actually live inside our phagocytes. So the actual cell you send to kill ends up ingesting the microbe and then protecting it, right? And we call those facultative intracellular pathogens. And probably the poster bacterium is Mycobacterium tuberculosis. Mycobacterium tuberculosis can live just fine in these phagocytes unless they become activated. And we'll see there's going to be chemical messengers that other cells of our immune system, and the one, you guys, the king of all the cells of the immune system are our T helper lymphocytes, our CD4 positive T cells. If our T helpers can supply chemical messengers to wake up this phagocyte, they'll turn it into an activated phagocyte. And activated phagocytes, you guys, are sometimes described as angry killers. They literally increase in size. They make more hydrolytic enzymes. They make more reactive oxygen intermediates. They can phagocytize invading microbes much faster and easier, right? So they're called angry killers. But the problem is, like in the very young, the very old, and people that are immunocompromised, and the tragedy is, we'll see in our members of our community that have HIV that's progressed to AIDS, their T helper lymphocytes are in such low numbers they can't activate these phagocytes, right? So if we can't activate the phagocytes, we can have really serious infections with these facultative intracellular pathogens like Mycobacterium tuberculosis, Salmonella, Shigella, Listeria monocytogene. I know I'm forgetting one. Yersinia pestis, right? So, yeah, moving on. So, you guys, so another interior nonspecific defense would be protection of fevers. And you might recall, you guys, from the medical micro unit when we were talking about bacterial endo and exotoxins, we said that substances that can trigger fevers are called pyro for fire, pyrogens, fire makers. And remember, you guys, we said that endotoxin is a pyrogen. As a class, bacterial exotoxins are not pyrogens as a class. So endotoxin is used as kind of the classic pyrogen. In the old days, we talked about it as being an exogenous pyrogen, a substance triggering a fever that comes from outside of our body. And we now know the mechanism is when the endotoxin is released, remember, it binds to cells of the immune system that release all of these cytokines, these chemical messengers. And some of these chemical messengers are going to circulate to the brain. I believe this is in the hypothalamus, prostaglandins are made, and that then triggers an increase in the setting of our internal thermostat. And you guys, I wouldn't ask this, but some of these endogenous pyrogens are tumor necrosis factor, interleukin 1, and interleukin 6, right? So then we will go through changes that will help increase our core body temperature. So we might start shaking. The muscle contractions generate heat in the periphery, we have vasoconstriction, right? So there's less blood that will move through the periphery, so less heat will be lost there. We'll have the sensation, the symptom of being cold, and so then we'll undergo behavior changes. So what we might do is maybe put on more clothes, or we might crawl into bed and put lots of blankets on, or we might turn up the thermostat in our house so the inside of the house becomes warmer, right? So all of this then is to contribute to an increased internal temperature. Now, why should that be helpful? Well, it turns out some microbes can't replicate well at high temperatures. So you might slow down pathogen replication. And folks, all the chemical reactions that we have or will discuss involved in defenses are going to speed up, right? You're going to have more collisions between those antimicrobial chemicals and the microbes themselves. The chemical reactions within the white blood cells are going to speed up, right? So they can make more antimicrobial compounds. So even though fever isn't pleasant, there's been natural selection for fever because it is part of our innate non-specific interior defenses. And is as Americans, we generally don't like to be uncomfortable, right? So usually whenever we start feeling bad, we wanna reach for a drug or something that will help us feel better. And so in general, folks, drugs that would reduce a fever or prevent it from being formed are called antipyretics. The oldest one that was known by indigenous people is aspirin, right? But then we have many generations of new antipyretics, ibuprofen and acetaminophen. And and folks, this might be something only if you're dealing with a healthy person, not a really young child or baby, and not somebody really old, and certainly not somebody who's immunocompromised. Some people argue that if you're relatively healthy and your fever isn't that high, maybe you shouldn't take antipyretics, right? Because if the fever is indeed helpful, maybe at inhibiting replication of the pathogens, maybe you're actually kind of you're defeating your own defenses, right? And trying to block that fever production. But again, folks, always be really, really careful um, with those high risk groups, little children, old folks, anybody immunocompromised. Um, when our kids were little, I was always on the phone to our healthcare providers whenever the kids went develop a fever, because I just wanted to make sure that it wasn't too extreme, right? So always be careful. Always call your healthcare provider if in doubt, right? And you guys are going to be my healthcare provider one of these days. I'll be calling you. So, folks, we just have two more interior innate defenses to go over. The complement system deserves a whole lecture by itself. So, let me see if I can't just kind of hit the key points here. So, the complement system, you guys, it's called a system because it's a collection of 30 different proteins. Most are made by our liver. So they circulate in our body fluids, blood, lymph fluid, extracellular fluids in an inactive state. And so you guys, we'll just use this little complement protein here. So some of the complement proteins, they have letter labels. Some of them have number labels. So here, this one is complement component 3. So when it's whole like this, it's inactive. For it to become activated, for it to become functional, it has to be split in two. So here we see we've got C3 being split into C3A and C3B. So it's these halves that are now functional. They take on their biological properties, right? So the way this works, there's actually 3 different ways to activate the complement cascade. You can do it non-specifically through complement binding to the surface of an invading microbe, and that can start the splitting process. That's called the alternative pathways. There's a mannose binding pathway, and then when we start talking about adaptive specific immunity, there is the classical pathway in which antibodies bind to invading microbe, complement binds the antibody, and the complement is cleaved. Okay, so we're not going to worry too much about the 3 different ways of activating complement, but what we do want to know is, how is it helpful? Why did it evolve? And oh, and one more thing, folks, it's called an amplifying cascade. So I think of a cascade like a waterfall. You know, once the water goes over the edge, it's going to go all the way to the bottom. Or you can think of dominoes, right? Once, if you line up dominoes standing upright and you you knock over one domino, it'll knock all the others over. So this is like the first domino, you knock them over, and all the other complement components are going to get activated. Amplifying because one complement component, when it gets activated, can activate the next complement component in the sequence. And maybe like this one active complement can activate 100 of the next complement components. And then when that gets activated, it can activate 100 of the next complement components. So that means you could start out with a tiny signal, just a few microbes invading. The complement is activated, and starting with that tiny, tiny signal, you can have a huge, huge result. Okay, so what's the result, you guys? Let's walk through this. This is something I would like you to know. You don't have to memorize the numbers here at all. This is called the terminal pathway, and this is where all the good stuff is made, okay? So what's made? So when complement is activated, we're going to make activated complement can act as inflammatory mediators, right? So here, and again, you guys, you don't have to memorize this. So C3b, C5b, they can bind to mast cells, for example, and trigger release of inflammatory mediators, which triggers all the benefits of inflammation. So remember, increased blood flow, we're going to attract white blood cells to the scene of invasion. We're going to guide them with the inflammatory mediators. We're going to activate them, right? So inflammatory mediators, um, um, activated complement can act as inflammatory mediators. Chemotactic factors, so activated complement can help guide the phagocytes to the scene of microbial invasion. Here's the opsonization, you guys. So complement can bind to the surface of some invading microbes where it will be activated, and then complement is then coats the surface of the microbe, so we call that opsonization. And here again, you don't have to memorize this, you guys. C3b is a opsonin. And then what's so cool is our phagocytic cells, our macrophages, our neutrophils, they have receptors for complement, right? So now that we have complement coating the microbe, now the phagocytes can really attach strongly and quickly ingest the opsonized pathogen, and that will increase phagocytic killing. And then this last one, you guys, is so cool. It's called the MAC attack, the membrane attack complex. So the remaining complement components in the terminal pathway here, when they're activated, they can insert into lipid bilayers. So they could insert into cell membranes of any cellular organism, causing these membrane attack complexes, these pores, right? So they can insert into cell membranes, they can insert into the outer membrane of gram-negative bacteria, they can insert into viral envelopes, right? So if this was a cell, if you have all these membrane attack complexes, all these holes in your membrane, you're going to lyse, right? So you can have direct lysis of cellular organisms, right? Or we could damage the viral envelope, and we know once we damage the viral envelope, it can't, it's no longer infectious, right? So the membrane attack complex, that's so cool. And this, you guys, this to me was just amazing. This is electron micrograph of the cell membrane of red blood cell. It looks like somebody's taken a machine gun, right, and just riddled the erythrocyte membrane. But what are all these little holes? Those are the membrane attack complexes. Isn't that wild? You guys, this is the last one for interior nonspecific defenses, and this is one where I wish I did have a whiteboard here, because this works well as a cartoon. It's like in steps, but let me see if I can do this right, you guys. Okay, so there are 3 different types of interferons that humans make: alpha, beta, and gamma. Gamma interferon we're going to describe in the adaptive specific immunity. We'll talk about it in cell-mediated immunity. So in non-specific interior defenses, you guys, we're going to only talk about alpha and beta interferons, right? And very often the interferons, they do abbreviations. They'll put INF alpha or INF beta, okay? So alpha and beta interferons can help protect us against viral infections. And the way the story goes, let's pretend that one of our cells has been infected by a virus. Now, if we're lucky, that infected cell will make alpha and or beta interferon. That's a chemical messenger. It's a chemical alarm. When we were growing up, if there was a fire somewhere, the local fire alarm would go off. The fire siren would go off. Well, interferon alpha and beta is kind of like a chemical fire alarm. It's telling neighboring cells there's bad guys, there's pathogens in the neighborhood. So get out your weapons, be prepared. You know, you're going to be attacked in your houses and your cells. So neighboring cells, in response to the binding of alpha and beta interferon, they'll start making these really cool antiviral proteins, AVPs, right? So this is like the neighboring cells. These are the weapons that they're pulling out, preparing to be invaded by these viruses. So that first cell that set out the chemical alarm, the alpha and beta interferon, that little cell might be killed by the viruses, right? But when the newly replicated viruses then come and bind to the neighboring cells already are awake. They're already activated. They're chock full of these antiviral proteins. So even if they're invaded by the viruses, the antiviral proteins will stop the viral replication. And these are just some of the mechanisms, you guys. Antiviral proteins, they can degrade the viral mRNA, they can inhibit the ribosome function, so none of the viral proteins can be made, right? So if this can happen early enough, you can stop the viral invasion, you can stop the viral infection. That's just amazing. So you guys, so that's the end of this innate nonspecific host defenses immunity. So we'll close this one up, and then the last audio we'll do will be on the second immunology PowerPoint on adaptive specific host defenses or immunity. And folks, we'll just go over um, just some really brief key points in specific adaptive immunity. Um, yeah, immunity deserves a whole course all by itself. So we'll just kind of give you some appetizers. And the other thing I'm just really glad about is that in your A&P course, you're going to get awesome immunology in your A&P course. So this will just be like the little appetizers if you haven't had A&P yet. And if you have had A&P already, a lot of former micro students said their A&P notes were really helpful. Okay, you guys, you take good care, and we'll be talking to you soon.